Welcome to Thinking Thoracic, the podcast dedicated to exploring the advancements in thoracic surgery. Brought to you by the Society of Thoracic Surgeons. This podcast features leading thoracic surgeons discussing and debating the latest research, surgical techniques, and trends to expand your knowledge, challenge your thinking, and help you stay ahead of the curve. Whether you're a thoracic surgeon, advanced practice provider, researcher, or industry professional, Thinking Thoracic is for you. Let's get started. The Society of Thoracic Surgeons Thinking Thoracic podcast is sponsored by AstraZeneca.
Hello, everyone, and welcome to Thinking Thoracic, an STS podcast. I'm your host, Erin Gillaspie, and it's my absolute pleasure to welcome you. I'm so excited to bring you the latest and greatest in thoracic surgery. Before we get started, a quick reminder from the Society of Thoracic Surgeons. You've probably seen the emails about the STS and EDGE research survey on non-small cell lung cancer. If you haven't had the chance to fill it out yet, there's still time. It only takes about 15 minutes, and your input will help the STS understand how evolving immuno and targeted therapies are shaping surgical practice. I'm so delighted to be joining you today with some amazing friends. I think one of the beautiful things about medicine is that we're all connected throughout training, mentors, colleagues, and the three of us are connected not only by our time together at Vanderbilt, but by the brilliant Otis Rickman. I think no podcast could be complete without mentioning him. He recruited, hired, trained all of us in different capacities. So big shout out before we get started to Dr. Otis Rickman. So now I'm going to jump in with some introductions for our two incredible guests that we have today. So first, Dr. Fabien Maldonado. He's a professor of medicine, thoracic surgery, and mechanical engineering at Vanderbilt. He's a brilliant clinical trialist conducting the VERITAS trial, which we'll be chatting about today. He's done tons of work with lung cancer, with the Vanderbilt Institute for Surgical Engineering, or VICE for short, helping to develop new robotic equipment. He is a brilliant friend and colleague, and as a side note, in his spare time, he has pursued a master's of science in philosophy. My friend, I'm just exhausted thinking about all the incredible things that you do. Welcome, Fabien.
Well, thanks for having me. I'm excited just listening to you.
Next up, we have Dr. Rob Lentz, associate professor of medicine in the Division of Allergy, Pulmonology, and Critical Care Medicine at Vanderbilt University Medical Center. He has a special focus on advanced diagnostics, complex airway management, and was also part of the lead investigator group for the VERITAS trial and within the Vanderbilt Interventional Pulmonology Research Group and the Pulmonology Outcomes Group. In his spare time, he's also the fellowship director for IP. Another brilliant friend, colleague. Gosh, Rob, you were my partner in crime for some of the most complex airway cases I did at my time in Vanderbilt. So I'm just so grateful that you're here today, too.
Yeah, thanks for having us, Erin.
All right, you guys, this trial, completely incredible. So excited that you agreed to be on to talk about the VERITAS trial. So for our listeners, this is really, in my opinion, an absolutely practice-changing trial within the space of IP. It was published in the New England Journal of Medicine. So let's dive right in. Fabien, I'm going to kick things off with you. Would you talk just a little bit about the background, inspiration, and really what was your impetus for doing this trial?
Yeah, sure, Erin. And again, thank you so much for having us. Really nice to talk to you again and miss the good days, both at Mayo and mostly at Vanderbilt. The VERITAS trial was really inspired by new technologies that we had available. Starting in 2018, as you well know, we do a lot of navigational bronchoscopy. And since 2004, when navigational bronchoscopy was first introduced on the market, we were always trying to chase after CT-guided biopsy that had the luxury of having intra-procedural CT guidance, which we never had. We had the good old, fluoroscopy, 2D imaging. And we based our planning on pre-op CT. And so you can imagine with the volume of the lungs almost at total lung capacity during the pre-op CT versus the volume of the lung of the patient on the table, intubated, paralyzed, and sedated, there's quite a bit of divergence there. What changed a lot is the ability to get 3D intraprocedural imaging to recalibrate the system, make sure the target is in its right location. And with that, we saw almost overnight a jump in diagnostic yield from about 50-50, which was where we were at the time, to about 75 to 80%, repeated in multiple retrospective and prospective single-arm studies. So armed with that knowledge, we went to knock on Medtronic's door and told them, you need to give us a lot of money for an investigator-initiated trial. We want to go head-to-head with your biggest competitor, which is CT-guided biopsy. And to their credit, they gave us a lot of funding. So this was received at the end of 2018. We had the protocol written up and the sites ready to go in 2019, and we started recruiting right during the COVID pandemic.
That was not an easy time to get things done by any stretch. And so especially kudos, because let's face it, a lot of trials sort of disappeared during that time period because we couldn't accrue. It was very, very difficult. So let's dig a little bit into the pathways for expedited technology approval. So one of the things that I've heard you talk a lot about in the past, Fabien, is there's different pathways that we can think about bringing new technologies onto the market, which has been extraordinary, right? And we've seen a huge boom in the last really decade in particular in IP. And there's a lot of really fun things happening, right? Electromagnetic navigation. We've now got robots. We have all of these different things. And there's been a lot of questions as we bring new technologies, new procedures on, that we're not always vetting them in the richest way, right? You know, for a long time, we jumped into CT guided biopsies because it's what we had. We didn't necessarily vet it to say it's the best option. It was just what we had. And we've sort of fallen into that trap a few times. Would you just expound just a little bit on approvals, some of your thoughts in that space?
Yeah, sure. No, that's a really good point to bring up. I think it's fair to say that most people don't realize that most of the technology we have dropped on our lab and that we start using on patients has never been demonstrated to improve patient outcomes in any way, shape, or form. And this is due to the fact that device regulation, medical device regulation, is fairly recent. It's a fairly young law or amendment to a law that dates back from the 1930s that regulated pharmaceuticals. So this is really late 70s, 1978, with the Device Amendment Act that introduced some kind of regulation for medical devices. And that regulation was really supposed to kind of mirror what was being done for drugs. And so if you had a drug that was exactly similar to something else, then fine, you can go ahead and commercialize it. But most of the time, you're going to need some randomized data to show demonstrable improved patient outcomes. And so the main pathway for device regulation in this Amendment Act of 1976, I think it is, was the pre-market approval pathway, which really required the same level of evidence that pharmaceuticals required for commercialization. Now, at the time, there was a recognition that with all these grandfathered devices already on the market, slight modification of these existing devices could not really go through complex, randomized, controlled trials. Plus, the device industry doesn't have the deep pockets that pharma does, so this was not possible. So that accessory pathway called the 510K pathway was created, and this allowed device manufacturers to commercialize devices based on their quote-unquote substantial equivalence to already commercialized device, so-called predicate devices. And what's happened over the past 50 years, 40 years, can't count, is...
1990 was just 10 years ago, right?
So 1978, so almost 50, right? I'm about 50 years old. What's happened is that this has really become the de facto pathway for medical devices to be cleared. So the FDA will look at a device and preclinical data and the request from the medical device company saying that this is really equivalent to that stuff that's already being in use. And then the panel will decide whether that's true or not. The problem is that the concept of substantial equivalence has never been really clearly defined. It is defined in terms of same intended use and technology, or if it changes, it doesn't pose new threats of risks to the patient. But that's a fairly loose and not well characterized definition. And so what's happened over time is that you start having devices that kind of evolve, like in a Darwinian kind of process, where you end up with a completely new organism that has nothing to do with the predicate device from 20 years ago. That predicate device may actually be withdrawn from the market because it was found to be dangerous, and it's still being used as the original predicate device. And that's true for all most devices that we use in interventional pulmonology, but it's also true of all medical devices in general, the Da Vinci robot, for example, right? So,
Yeah.
What that's done in practice is that it's really shifted the responsibility of producing data from the device manufacturer, which, to be fair, don't usually have the resources to run these clinical trials. The NIH is not particularly interested in picking that up because that's not their responsibility. And so it falls upon us to do these trials. The problem is funding, regulations, difficult. And so that's why we're so proud of these trials, that we get it done, and that really spearheaded a movement, I think, in our little niche specialty of randomized control trials that are really exciting.
I agree. I think it's really extraordinary. And you really pushed the envelope and said, hey, guys, we need to make sure that we're providing patients with the best outcomes across the board and making sure we're using our technology to the best of our ability. So massive, massive kudos.
Thank you.
All right, Rob, you're up. Take me through the clinical design for this trial. How did you choose a sort of nodule size, nodule location? Talk to me about randomization and how did you choose your outcomes?
Great question. So we spent a lot of time on designing this with a few important things in mind. So the problem with the lung nodule biopsy literature since we began biopsying lung nodules is that almost all the literature out there were single center, mostly retrospective, and unique, all non-comparative studies, right? And so studies that produce highly biased estimates of how good these, and that's true for transthoracic needle biopsy. That's true for all of our bronchoscopy studies as well. These are all single arm trials. So the literature is biased. And so we designed this trial specifically to try to limit, develop robust evidence, limit the ability for bias to seep in. And largely that's in the form of selection bias, right? So if you only go after nodules, clinically you get a message, can you go after this nodule? And I think it's 50-50. Why don't you call IR instead of us? And they do the same thing, right? And then you publish your series of biopsies and, I'm 95% diagnostic yield because I only go after what I think I'm going to get, right? And so that was the major thing that we had to contend with in designing. How do we prevent selection bias from seeping into this trial? And then the other thing, kind of in a related fashion, is we also couldn't ethically randomize patients to a biopsy modality that was either going to be unsafe or they had no chance of getting a diagnosis, that the biopsy was not at all technically feasible. And so we had a few built-in, we developed a central adjudication pathway or central review pathway. So patients were enrolled, consented, and then a central panel of interventional radiologists, separate from a separate panel of central panel of interventional pulmonologists, reviewed these scans that were sent centrally. And within 3 days, usually within 1 day, each group reviewed the scans and said, yes, this is a technically amenable case. And if it was not, you had to declare why. And we had some preset, like for IR, it was they had to cross multiple fissures, they had to cross a bolus, fairly straightforward exclusions for them. For us, it was no airway within, I think we said 3 centimeters, no visible airway within 3 centimeters of nodule, which essentially never happened. So that was one way we kind of built in. So your cards were on the table, and you had to show your work, and you had to defend. You had to defend if you said this was not technically amenable. We tracked it and we recorded that. So that was part of it. And then to avoid selection bias in the nodules that came to us, we partnered. Each site partnered IP and IR. We embedded in both referral streams. So these weren't just, the types of nodules that go to both places might be different, probably are in some way. And so we embedded in both. So all those nodules were reviewed. It wasn't just nodules coming to pulmonary that ended up in the trial to make sure we weren't getting a biased sample there. Once we declared this was amenable by both groups, then that's when randomization happens. And then we went on to do the biopsies. Randomization was 1-to-1 between the two arms. Other important inclusion, exclusion, nodule size. So these are 2 nodules, 10 to 30 millimeters. These days we're going after things routinely smaller than 10. I'd say, and we shouldn't go after every 5 millimeter nodule, but that's in the realm of possibility now with what we're doing. 10 was chosen because that's actually in the FDA approval for the device we use, the navigational bronchoscopy device we use, which is a limb site. So that was the decision on 10 to 30, but we did not include masses, right? So these were true lung nodules. The median nodule size ended up being pretty small at 15 millimeters. We built in, most of these nodules we're going after because we're concerned for cancer. They all had to have at least a 10% pretest probability of malignancy. Outcomes, the primary outcome was diagnostic accuracy. So that is, this has been an evolving thing in our field too, right? So in all of our papers, you'll find the words diagnostic yield. This was the diagnostic yield. This procedure was whatever. But in almost all of the studies prior to this, before we started thinking about our outcomes here, basically we're saying we got a result and we followed the patient for 1 to 2 years, and we didn't miss any cancers, right? We're basically measuring sensitivity for cancer in many of these studies and kind of ignoring benign disease. And so more recently, we've developed a more robust set of criteria for what actually constitutes diagnostic biopsy. And so when you do a lung nodule biopsy these days, for research purposes, you can put it into one of a few buckets. If it's cancer, we assume it's diagnostic. It almost always is. It's malignant. If it's benign, there's different flavors of that. It can be specific benign, such that it's a pathologic pattern that it comes with a specific diagnosis. So hamartoma, amyloidoma, granulomatous inflammation, something that highly predicts and explains a lung nodule. In the study, we still followed those over time to make sure that they behaved benign, that they didn't end up having malignant diagnosis. And so we caught false negatives in that manner. And that was the outcome. We had to have a diagnostic procedure. So we had the positive diagnostic yield, a diagnostic upfront procedure based on these conservative criteria. And then we followed for at least 12 months and to make sure that we were not missing any false negative benign biopsies. And that gave us our primary outcome, which was diagnostic accuracy at 12 months.
I love that. That's been a huge change because it's sort of been previously very nebulous how you can feel confident with a negative biopsy, right? Does it have to have a granuloma to feel confident saying, hey, it's not cancer? Does it have to, what if you get something that looks like fibrosis, like scar? There can be some very non-specific findings, more consistent with ILD and some of the other changes we can see in lungs. And so having that really robust definition, it's nice to have that now, and it's nice that it's going to be carried forward now as sort of a baseline for all future studies too.
Yeah, this is something we kind of ignored and didn't think carefully about for years. And then as we got into more robust trials and we started really designing these and trying to be as robust as possible, we started thinking about these outcomes and realized that what we were doing. So not only were all these single arm trials prior to this on both sides, IP and IR, right? Not only there's lots of selection bias, there's publication bias, there's lots of biases up front. And then all the outcomes are wildly different because there's lots of different ways you can kind of massage these outcomes in a not strict conservative diagnostic yield way. So we have, I think, finally seen the light. And Fabien was just on we have an ATS ACCP consensus statement now for the appropriate research endpoints in lung nodule diagnostic studies that kind of codifies. And it hopefully will be an evolving document as we get more data, as we validate this definition, as we find, atypia suspicious for malignancy is almost always malignant. Maybe that's diagnostic. Currently, it's not. Or necrosis without atypia is almost never cancer. Maybe eventually that becomes a diagnostic signature. It's not currently. So hopefully this will continue to evolve as we do more data, do more research, and get more data on and kind of validate this definition.
I love that. So you had basically nodules in the peripheral third of the lungs, 10 to 30
peripheral
millimeters.
Peripheral two-thirds. So inner third was out for two reasons. It's a riskier and a longer shot and a lower yield for IR. They don't tend clinically to often go after those. Some sites may. And many of those are accessible to much more straightforward and safe bronchoscopic methods. So either white light bronchoscopy, we can see it in the airway, or EVIS, ultrasound guided bronchoscopy. So yeah, outer 2/3, and we stratified outer third, middle third in our randomization.
Perfect. Love that. So you have outer third, middle third, 10 to 30 millimeters, solid nodules only, and at minimum a 10% predicted risk of malignancy.
Yeah,
Love
and
it.
it was actually all comers. It was not just solid. It was Mostly solid. It was about 80% solid, 16-ish% heart solid, and about 5% pure GGOs.
Your GGOs. Beautiful. Now, randomized 1-to-1, that's incredible. You guys accrued, finished the trial, which is also incredible, especially, during that time period. But the interesting thing is also during the time period that you guys were running your trial, there was a lot of new technologies coming out, improved navigation, obviously robots, smaller cryoprobes. And one of the questions a lot of people have when they look at this trial is, how did you guys standardize, or did you standardize approach to biopsy, or did you give each person doing those biopsies the leverage to do what they think is going to be the best option to get the highest yield? And then if so, what were the dominant approaches for this study?
Yeah. So we did not, we were cognizant of avoiding over protocolization. We wanted to make this as generalizable as possible in the context of an explanatory randomized controlled trial.
Yeah.
And so starting with 7 sites across both academic medical centers, community hospitals, across wide geography in the United States. So trying to get as representative and generalizable sample as we can. Procedures themselves. So we did standardize the navigational platform used, which was Lumicite. So it's digital tomosynthesis guided electromagnetic navigational bronchoscopy. Beyond that, we did require that the needle, you take at least 3 needle passes. And that was a practice at all the sites we used. That was not changing anything that people were doing. That was standard at all sites anyway. We did not, I don't think we actually protocolized the use of ROSE, but in all bronchoscopies, ROSE was used. ROSE was also available, interestingly, for, all these hospitals had ROSE. It was available for transthoracic biopsy as well, but was not utilized very often in that arm by operator choice. Everything else was up to the operator, whether you did digital tomosynthesis or not, whether you used additional tools, how many spins you did if you did DT. All of those things were up to the proceduralist, and we didn't dictate any of that. And we also didn't dictate anything that happened afterwards. All we did was follow the patients through at least a 12-month CT scan, if we were following a benign lesion to confirm what it was in the end, cancer or not.
No, I love that. Actually, I really appreciate that you guys didn't standardize it, right? Because it allows the clinical flexibility to make safe decisions. It allows a study to grow with technology as it continues to expand. And so I think that's really, really extraordinary.
Yeah, and I will say, so cryoprobes are our favorite tool these days.
Yes.
They were not used. We finished accrual kind of before the cryo wave crested. So it was not. It was top of my head, under 10%, maybe under 5% of cases in the study used a cryoprobe. So this data does not really reflect the cryoprobe craze that currently has washed over all of us.
STS surgeon members, we'd like to hear about your experience treating patients with non-small cell lung cancer. If you're a general thoracic surgeon and see patients with non-small cell lung cancer, you should have received a survey in your inbox. Please look for that email and take 15 minutes to complete the survey. Your input will help STS understand how evolving immuno and targeted therapies are shaping surgical practice. Please complete the survey by November 3rd, 2025. If you have questions, contact STS's survey partner, Edge Research, by emailing frank@edgeresearch.com. Thank you for your help.
Now, you guys designed this as a non-inferiority trial. Why did you choose non-inferiority as opposed to superiority? I think it's always really important for our listeners to sort of hear about the different opportunities when you're thinking about designing a clinical trial and some of the decisions behind that.
Maybe.
Yeah. Right. So first of all, one of the assumptions on which we designed the trial was that CT-guided biopsy was accurate 90+% of the time, if not 95%. In fact, we designed VERITAS 2, and some of our interventional radiology colleagues said 95% is a bit low for me. And so that we start from there. It's really hard to power for an effect size of 2% or 3%, right? But importantly, we had no illusion that we would be better than CT-guided biopsy. What we want it to be is close enough, because we have with bronchoscopy non-efficacy benefit that justify non-inferiority design study. So for example, the complication rates have been clear from the literature for many, many decades now that navigational bronchoscopy is less risky in terms of pneumothoraces and so on. But it's not just that, it's also the ability to sample the mediastinum in the same setting with EBUS, the ability to biopsy multiple nodules, which is increasingly something we do. And so for all these reasons, I think a non-inferiority study design made a lot of sense. We had assumptions that were derived from the literature. So we were going to be 80%. That's what we had been publishing with that particular platform. And oh, by the way, to your point, Erin, it's like we started this study and 6 months later we had a robot, and then there was another robot. There's 3 robots on the market, and then you've got cone beam CTs. And it's made it hard, right? Because we are trying to recruit patients, the collaborating centers, we got the robot. Everybody wants to use the robot. And so you have to convince, people that this is really an important thing to do. But that gets back to the equipoise question, right? Which is really suspending your belief for the purpose of the greater good, which is hard. As physicians, we don't like that. We don't like to acknowledge uncertainty, and patients don't like to acknowledge uncertainty. And so many patients were like, nope, I want the robot because I've heard about it, I've seen the billboard, I've seen the LinkedIn posts, etc. So going back to your question, though, non-inferiority made a lot of sense because we had, on the table additional non-efficacy benefit that we could, argue for in the context of bronchoscopy.
I love that. Well, you've alluded to maybe some of the results we can expect. Rob, hit us with the results. What did you guys find?
So top line headline here is the navigation bronchoscopy was non-inferior. We met our threshold for non-inferiority. So NavBronch, non-inferior to transthoracic needle biopsy for true lung nodules amenable to both procedures. That is essentially the inclusion in summary. The actual diagnostic yield in navigation bronchoscopy, accuracy, primary outcome diagnostic accuracy, 79% for bronch, 74% for transthoracic needle biopsy. Diagnostic yield, not accounting for follow-up, was 79% versus 78%. There were 4 false negatives in the transthoracic arm, which made their accuracy drop a few percentage points. So that is the top-down or the headline on the primary outcome and the main secondary outcome, which was yield. And then complication rates. Overall, any complication was about 29% in transthoracic biopsy versus 5% for navigation bronchoscopy. Now, to be fair, most of those in the transthoracic arm were pneumothoraces that were small. And of course, these patients are all, by definition, underneath the CT scanner. And they all have a CT scan right after the biopsy is done, and any trace little pneumothorax, ding that bell. And so when we look only at those that are really clinically significant pneumothoraces, there still was a significant difference. So it was 12% of pneumothoraces in the transthoracic arm needed hospitalization and or chest tube versus 1%, just under 1% in the navigational bronchoscopy arm. So we conclude from this then that navigational bronchoscopy is not inferior. That's what we can conclude based on our design. The actual diagnostic rates were very similar, and navigational bronchoscopy is much safer, much fewer complications.
That's the conclusion. I think it's important also to point out that these are for nodules that have been judged to be amenable to either or procedure. There are still nodules that are clearly better served by CT-guided biopsy and some that are better served by navigational bronchoscopy. So if you look at the number of patients who were enrolled over a period of 33 months, we screened 519 patients. Well, that's the number of navigational bronchoscopy we do in a year at Vanderbilt, right? So at 7 centers. So it tells you something about how refined that population of patients was. And this also speaks to the strict eligibility criteria for the study. So as Rob pointed out, we actually, initially, my dream was to have a more pragmatic type study. And that's where you see the flexibility of co-intervention. Yes, we mandate some things like TBNAs, but the use of ROSE is at your discretion, and the use of cryobiopsy, forceps biopsy, that's really entirely up to you. So we integrated some pragmatic clinical trial component to this study, but it turned out to be quite complicated and quite an explanatory type trial, which are actually quite useful when we submitted the paper to the New England Journal of Medicine, because that's what they want. And so that was really, really great. But in retrospect, some of the things that we did perhaps could have been loosened a little bit. So Rob was talking about the stratified randomization from middle and outer third, for example. That sounds great in theory, but in practice, when we set out, oh, how do we do that? So now we need to get CTs from all these centers, and we need to have some kind of HIPAA-compliant cloud-based platform that didn't really exist at the time, that we had to create for the purpose of the study, that thanks to some Vanderbilt colleagues were able to do. And then you get to sit down for the 288 consented patients. And I had to manually segment all these lungs and figure out where the nodule was located. So an enormous amount of work, which in retrospect, maybe we don't need to do all these things. So they are, by virtue of this being a very much explanatory type of trial, but also by virtue of trying to be fair to our interventional radiology colleagues, we captured a narrower population of patients that we serve every day, with bronchoscopy. So with that caveat in mind, yes, we are non-inferior to CT-guided biopsy.
Yeah,
I think we're much better than CT-guided biopsy. But
I'll make the case though. So if you drill down on the exclusions and kind of how this all flowed, and if you really want to dig on this, I have a Sankey diagram in the supplement that it took weeks out of my life, and it's beautiful. And I don't know if anyone's ever seen it, so go look at it. I do think this was a pretty generalizable result, though. So Fabien's right. So we screened a little over 500, and about half consented and half didn't. Of those who were excluded, about a third just declined to participate. Maybe they were different, maybe they weren't. And of those that met some exclusion criterion, which I'm not going to go through all of them here, the majority of them were because they had an abnormal mediastinum and they had a clear indication for bronchoscopy separate from their nodule. So that was most of the folks who did not enroll in the trial for fairly legitimate reasons. And then of those who were consented and went through this adjudication process for eligibility for both procedures, 18 were declined by IR. So a relatively small percentage of that number, actually. Only one was felt not amenable. That was also felt not amenable for transthoracic biopsy, case of obesity. So I do think that this is a relatively generalizable sample. I think we accomplished our goal of trying to really avoid selection bias and cherry picking on either side and on lung nodules of a whole. And I think this fairly well represents what these two techniques can do in a pretty generalizable cohort of nodules.
Particularly with the central adjudication, right? And having that many layers of re-review, everybody agreeing ahead of time that this is an accomplishable biopsy, really taking those steps and then giving everybody the ability to choose their best mechanism to do a biopsy. I agree. I think it's a very generalizable result and a really thoughtful way of interpreting and concluding all of the data, right? And so you said, hey, this is equivalent. So in two patients that have an equal opportunity to get CT guided versus endobronchial, we have an opportunity to have fewer complications. We have the ability to concomitantly stage the mediastinum. And so when there's equivalence, this is the opportunity that we should choose for our patients. And I think that's a really, wonderful conclusion to this paper.
Yeah, and the question that always comes up at the end of this beautiful conclusion that you just mentioned is, well, what about cost effectiveness? Because after all, navigational bronchoscopy is done in the operating room usually, or procedure suite with general anesthetic capacity. The patient is paralyzed. I mean, there are costs that you don't incur with CT-guided biopsy usually. But that is actually balanced by complications and taking care of complications sometimes require hospitalizations, about 11% in the CT-guided biopsy arm. One of our fellows presented the cost effectiveness analysis based on VERITAS last year at ATS. The paper is being published, and it's about a wash. So yes, it costs a lot more per case, but in reality, when you take into account complications, it actually kind of balance itself out.
Oh, that's very, very interesting. I'm really glad you mentioned about the cost analysis because I hadn't even really thought about sort of the added cost. You in your mind immediately, gosh, CT scanner is very expensive, and just all procedural suite time is expensive. And so it's interesting that actually even the space outside of the OR, those added pieces are the equalizer. Very, very interesting. So you guys are doing a great job. High level of diagnostic accuracy, keeping patients safer. Obviously, we've alluded to the fact a lot of new technologies have come out sort of during the time period and since the time period. What are the things that you guys think are going to drive this diagnostic accuracy even higher? Because I'm going to be honest with you, I thought it was fantastic, but one of the things that I hear is a lot of surprise amongst interventional radiologists. They all thought it was going to be a lot higher on their end. So they were quite shocked that it was, closer to that 80% mark. Do you think we're still sitting at about 80% diagnostic accuracy, or do you think this has actually continued to shift? And are we, we hear some people with the cryo, with the cone beam, with all the things, maybe saying we're starting to eke into the more than 90% with our endobronchial approaches.
Yeah, that's a really good question, Erin. I think that's where in a non-comparative setting, you're going to have to take into consideration what it is you're trying to accomplish, right? I mean, it's um, the diagnostic yield is an aggregate measure that captures so many different things, right? The environment in which you work, access to different kinds of biopsies, PET scan, how good your pathologist is, or specific or sensitive they are, what kind of adjunct technology you have. And to me, the diagnostic yield and accuracy, as it is reported by individuals or in studies that are non-comparative, is a reflection of how good you are at selecting patients that you believe you will be able to provide service to and benefit to. So the optimal diagnostic yield, I think, depends on who you are and where you practice and what population of patient you serve. It might be that you should be around 65% if you live with no access to biopsy other than what you're doing, and the alternative to that is resection. Then perhaps
Yeah.
65 is a good number. And maybe if you were 80 or 85%, you would not be doing enough. Uh, Conversely, if you have access to everything and you get a robot and great pathologist, 90 to 95% might be the right number. So I think that number is tricky. I do think we've hit a ceiling, probably. I can't imagine we're going to get much better than we are. I would say that the future of technological innovation in the interventional pulmonary space is probably going to be in the field of therapeutics.
Yeah.
And importantly, we're going to hit the same kind of issues magnified, because now we're talking about taking care of patients from a therapeutic standpoint. We're going to have the same issues of generating data that's going to be able to inform patient care with devices that have been cleared by the FDA for use. And we already have some of these on the market, and we already have people using these things, and no randomized data to date or forthcoming. And so that's going to be the next challenge for our specialty, I think, and for the future of our specialty, being able to secure funding for these studies, being able to follow some kind of principled pathway towards evidence generation that mirrors what's being done in oncology, for example, is going to be absolutely critical. And we can't, by the way, one of the arguments that are often advanced when we talk about these things, like, well, look, surgery didn't have any randomized data. Well, SBRT didn't have any randomized data. Well, true, but nothing else. I mean, you had a cancer, we needed to take it out. What else are you going to do? And then if you're not a surgical candidate, then you get SBRT. What else are you going to do? You had to do something. We're not in that situation anymore with bronchoscopy, ablation of peripheral lung cancer. We have SBRT. We have minimally invasive surgical intervention. And so I think the bar we have to clear for evidence is much higher.
Amazing comments, both of you guys. Thank you so much. So post Veritas, what are you guys most excited about? What do you have in the pipelines next that you're excited about from a research standpoint? Share all of the wonderful news with us.
Oh, we have so much, and we're so excited about this. So we've learned a lot doing Veritas, as Rob mentioned. I mean, this was an explanatory type of trial that was really well designed, but quite complex to run. And as we were running the trial, both Rob and I were getting so envious and jealous looking at our critical care colleagues doing 10,000 patient studies in half the time. And so we went to knock on their door and asked them to help us design more pragmatic type of studies. And we were able to do that. So at about the same time, VERITAS came out, Reliant came out. Reliant was a randomized control trial. It was the first cluster randomized trial in navigational bronchoscopy or interventional pulmonology in general, comparing the Ion robotic platform to the Illumicide platform that was studied in VERITAS. And that study was a single center study, but enrolled 411 patients in about a year. Absolutely amazing. We're so excited about this. We're able then to do Reliant 2 that just finished enrolling patients about 2-3 weeks ago, enrolling 440 patients at 2 centers. Now we'll get multi-center study using the same type of pragmatic clinical trial design. And I think that's the way to go. I think everybody's picking up the idea that we can actually have this almost kind of learning healthcare system where every patient we take for a procedure that's exciting and promising, but not yet validated, can be part of a trial that will help generating randomized evidence and inform patient care now. And that's a good thing because it protects patient now in the sense that each patient is given an equal opportunity for the so-called validated procedure and the new sexy, exciting procedure, and of course, inform the care of patients in the future as well.
It's amazing. Any closing thoughts, Rob?
No, I actually I drilled down on this, and Reliant enrolled 15 times faster than VERITAS, with I think under 5% of patients declined to participate versus about, we had about 50% screened that didn't enroll in VERITAS. So I agree, this is clearly the way as we keep, the pace of technology coming out keeps accelerating. We keep getting further and further behind on knowing what our devices can do. The only chance we have is to build research networks and push pragmatic trial design in a multi-center fashion. And if we do that, we can have, and combined with an outcome, diagnostic yield that's immediately adjudicated, does not require follow-up, right? We can have meaningful, robust, highly accurate results inside of 6 months. The DUAs are going to take longer, which is a nice place to be. I mean, I wish the DUAs were faster too, but still.
Amazing. Thank you guys so much for your time today. Thank you for all of your kindness and collaboration with me over the years. Congratulations on all of these trials, and I cannot wait to continue to follow all the incredible things that you're both doing.
Thank you, Erin.
Thanks, Erin.
To our amazing listeners, thank you so much for joining us today. If you have enjoyed the conversation, please be sure to follow Thinking Thoracic on your favorite podcast platform so you never miss an episode. Your support helps us to continue to grow, improve, and to bring you the most insightful conversations. And if you have a moment, we'd love for you to leave a rating or review. It really makes a difference. Thanks for tuning in, and we'll see you next time.
Thank you for joining us on this episode of Thinking Thoracic. The opinions expressed in this podcast are those of the participants and do not necessarily reflect the views of the Society of Thoracic Surgeons.